distribution. Okay, welcome everyone. Thanks for being at my presentation. And thank you for letting me be part of this session. This is my first time presenting anything related to microbiome or animals whatsoever. So I'm really excited to be part of this community, and I hope that I can have some really great conversations to get to know all of your work better too. Okay, here goes. So I'm going to be talking about valley oaks, the leaf and bulk soil parabiomes, so not just microbiomes, but parabiomes, that are partially shaped by host genetics. So the overview of what I'm going to present today is I'm going to first tell you what I'm calling the parabiome. I'm not sure how frequently this term is used. We'll go through how we do metabarcoding, lab work, and analysis. I've been talking to several students here that seem to maybe be interested in some of the methods, so I'm focusing on those a little bit heavier than on the results right now. And the state, I'll talk a little bit about the state of thinking of biodiversity distribution that we can track with parabiomes. I'll introduce then the Valley of Common Garden and the sampling design for this study. And then I'll show you some results on how season, site, being under oaks, and the oak population relate to the parabiome. And talk a little bit about what it means, maybe ask you some questions about what it means. And I'll introduce also on the side an eDNA, environmental DNA parabiome project that we have across California. That's a huge citizen science project that we launched last year. And that's it. So what is the parabiome? It's anything that leaves a DNA signature in the environment. You can collect from soil or from sediment and just put it in a tube, sequence it, and whatever you get thousands of DNA signatures that could be from different organisms that might indicate even population level differences. And as, of course, as sequencing is getting much more sophisticated, we can start to maybe track evolution in action or understand how populations are shifting with climate change with non-invasive sampling. So it's a really cool, promising technique. And when you're looking at more than the microbiome, there was no really good word for it, because it's not a whole biome, because you're not talking about they're functioning together, you're just saying something is nearby, so parabiome. Cool. And so a term that I might end up using is ASV, or amplicon sequence variant. So if you're doing PCR and you get two different products, maybe there's only one SNP that differentiates them, they each become a separate ASV. So from one sample, you get an average from a surface soil sample in California, we get about 1,000 unique ASVs per marker. So it's a lot of data. So we do metabarcoding, so we take different primers, we do separate PCRs using a non-GC biased polymerase, and we do separate PCRs because, of course, there's bias in your PCR, and then we pull them together and we clean them, and then we index them and do a standard sequencing. Make sure that we have the same number of copies, because that's always an issue. You don't want 10 times more of one marker than another. And then we sequence it on a MySeq, the 2x300. So so that gives us like a lot of contigs, which is nice. And we aim for 25,000 reads per barcoding locus. And I can look at my computer, I don't have to look up here. Okay. We get the FASTQ file stack, and then we process data with our own custom software pipeline called Anacapa, which is named after Mirage. It means Mirage Island in Chumash, which is sort of Mirage is if you're working with eDNA and metabarcoding, whether you're seeing a pattern that's real or not, or some bias because of your filtering standards, it kind of is an appropriate name, we think. Then you organize your metadata, and we have a data exploration tool I'm going to introduce to you called Ranacapa, which is just Anacapa plus the R for it's in R. And that's basically a phyloseq wrapper. Anyone in here use phyloseq? Okay, so be familiar, see some nods, great. You do some statistical tests, look at patterns, and then you stand up back dumbfounded by your results. And I've just been sort of like trying to interpret this. This is my first microbiome project. So the first step of Anacapa is you have to make a database, and everyone should get a database. Because maybe your primers are different, so you want a custom database for the exact primers that you're using, and maybe you want to filter based on geography. Maybe you want to filter based on having certain sequence similarity parameters, certain qualities. And public databases are great, The Silva Green Genes database has been tremendously valuable. There's an ITS2 database that's managed by Würzburg. I think it's the last name of the author. It's in Germany. But right now, with so much more sequence data being published all the time, you want to be able to have an updated reference database. And so we made one called a generator called Crux. It's really easy to install on your HPC. It's like there's like a research environment that you can just do it really easily. So if you're interested, talk to me about it. Then we do sequence QC and we do ASV parsing. So a lot of software that exists, they will only use the merged pairs. So when your forward and reverse reads make a contig. If you have like a reverse read alone, it doesn't get analyzed. It's wasted data that you've generated. Well, we don't waste anything. We use it all. And we're using programs like Data2, Botany2, a lot borrowing from the transcriptome community to be able to do that. And then we're using BLCA, Bayesian least common ancestor, as a way of assigning taxonomy, where you actually have a confidence value for every level of the taxonomic hierarchy. And so BLCA works pretty well, and so we just modified it to take in Botany2 results instead of blast results only. But yeah, so all of this is currently under review in Molecular Ecology Resources. Hopefully it'll be published soon. And it's already available on GitHub. So and if you want to use this, just talk to me. It's like pretty awesome stuff. So now switching gears into biodiversity distribution. We know that the landscape shapes biodiversity. And so we should not expect all sites that we visit to have the same biodiversity distribution of organisms from microbes to mammals, right? And so we also know that environmental adaptations in the host should affect biodiversity. If you are a healthy plant or a non-healthy plant, you might have a different community that can live on you or can use your resources. And there's been some really exciting work in the last couple of years showing not only do plants affect the microbiome community structure, but also some of their functional abilities. So here, this is a study on eucalyptus, where eucalyptus from temperate, subtropical, or tropical regions were actually influencing the temperature tolerance of the microbes in the soil. Just came out, so we put in. Okay, so now I'm going to tell you about the actual project that you're here to learn about, the Valley Oak Common Gardens in Chico and Placerville project has actually been started several years ago by Jessica Wright, who's here in the room, and Victoria Sork. And if there are others that I'm missing that went into the initial design, tell them that. Okay, so this is a two-site provenance trial. And they gathered seeds from 95 different populations across California, which in California, biodiversity hotspot, I don't need to tell you all that, but each population seems to have its own genetic group. They germinated many seeds from each provenance in 2012 in a greenhouse, and then planted them outside in 2015. In 2015, someone else, I wasn't there at the time, but someone else had collected soil from between the trees that were being planted at 10 centimeters depth and at 50 centimeters depth. So in 2017, when I started working on this project, I repeated that same sampling from those same spots at those two depths.